I see Renee has joined. Hi, Renee. How are you?
Very well, thank you.
Yeah. So, either you or Eric can start, whichever one you'd like. So basically, everyone, welcome. This is our Department of Psychiatry meet and greet for psychiatry and neuroscience research training programs. These are our T32 and our R25 funded research track during residency. And we have our chair of psychiatry, Renee Kahn, and Eric Nestler, the director of the Friedman Brain Institute, who will give a brief introduction about brain research at Mount Sinai. So who wants to go first? Oh, I think you're muted, Eric.
I am muted. Sorry, Renee, you should go first.
Oh, okay. All right, then. Thanks very much, Eric. Thanks, Mercedes and Antonia. Yeah, I'm very happy to welcome you for a program that is really unique and has a very long tradition. What I usually say is that I'm probably the first, although unofficial, graduate from this program. I'm not going to tell you when this graduation was because it was a long time ago. But anyway, it was one of the first graduates probably because this program was started by the current CEO of the whole Mount Sinai system, because at that point he was chair of psychiatry, Ken Davis, and I trained here as a resident and at the same time did a research program. So we have a very long tradition, a tradition of about 30 years in doing this program, and it has been incredibly successful. Many of those who went through this program as MD PhDs or MD researchers have continued to go into the faculty, including myself, obviously. And Mercedes is one of them, and we've got many others. So they've stayed here in our faculty or went elsewhere and have really made very important contributions into neuroscience and psychiatry, both clinically and basic science. So we have a long tradition. It's very well supported, and like I just said, it couldn't be more in safer hands because, like I said, the CEO of the system actually started this program. So that's always important when you start a program. How well embedded is it in the system, and how well supported is it by the leadership? And both are, very strong positives. We're very well embedded, and we're very highly supported. And obviously, I support it very much because, that should be very clear. I went through it. I've been helped very much by it. And I think it's really important. And why do I think it's important? Because it provides psychiatrists, doctors, with the possibility to combine it during their residency program with doing research. And that's a very privileged situation that we have, that we can both, treat our patients, see our patients, understand what the illnesses are that we are studying, and at the same time develop ourselves into researchers. And that researchers can be from all the way really basic science in doing genetic studies up to doing clinical trials in whatever disorder has your fancy. Because that's what we do. We have a very large program in disease from autism to Alzheimer's. We cover the entire field in disorders in psychiatry. And same thing holds true for the kind of research we do, from really stem cell research up to large clinical trials in, for instance, depression, developing new drugs. So it's the whole spectrum in disorders, and it's the whole spectrum in methodology that you could use. So it gives you a lot of choice. It gives you a lot of possibilities where you want to go and where you want to develop yourself in. We have a very good mentorship program. We mentor the trainees, obviously, but we also mentor them in providing help for getting new grants out, K awards, for instance. We have a T32 program, which is not relevant at this point in your career, but it's helpful in having people who finish this program bridge their time until they have their first grants out. And that also is very helpful in providing support for scientific careers. Like I said, we have a proven track record that the people who went through this program are, and you probably meet a lot of them here, have been very successful and have been really helped by this program. So that it gives you a little bit of an idea what we have. We have been doing this for many years. It works out well. I'm also quite personally involved in mentoring, and so are many others. Actually, that was for junior faculty. I went over 3R1 applications this morning with candidates and giving them my pretty direct comments on that. Anyways, so that is our tradition. As a research faculty, we're doing extremely well. The latest numbers are that 36% of the NIH grant that we submit actually get funded, which is a huge, very, very high percentage. As a department, apart from medicine, which is the largest department in the school, we submit the largest number of grants of any department, basic science or clinical. I mean, sorry to brag, Eric, but you know, it's the purpose is good, I hope. So we turn out a lot of grants and we get a lot funded, which is great. So I think you are in an environment that is really doing well. Finally, it's a highly collaborative environment. We really work very closely within the Friedman Brain Institute, and Eric Nestler is director of that institute. We work very closely with other departments, most closely with neuroscience and genetics and imaging. And sometimes it's, people have dual appointments, and I don't even know whether primary position is in neuroscience or in genetics or in psychiatry. That's how closely we work together. And that's really good. And I can tell you that is certainly not the case in all the departments that I know of. So let me end there. I think this gives you a little bit of overview, and free to answer any questions later on if you have them.
Thank you, Renee. So Eric.
Sure, no, thanks, Renee. That was a fantastic overview. So I'm Eric Nestler. I'm director of the Friedman Brain Institute and also a psychiatrist. This program is extremely important to all of us, Renee and me personally, but to Mount Sinai overall. Renee referred to our leadership, I'm Dean for Academic Affairs. Our overall Dean is Dennis Charney, also a psychiatrist. Our CEO is Ken Davis, also a psychiatrist. Psychiatry is uniquely strong here at Mount Sinai. What we have tried to do over the years is to build a neuroscience community that is uniquely integrated, as Renee alluded to. Perhaps Renee and I imagined a few decades ago that the fields of psychiatry and neurology might be merged at some point into a residency training in clinical neuroscience. I don't know if we're any closer to that now than we were 30 years ago. We can debate the values of that or not, but what we have done at Mount Sinai is a functional integration across those two departments, but adding neurosurgery, basic neuroscience, ophthalmology, rehab medicine, among many others. So we have tried to break down the barriers that sometimes exist between these individual departments to the detriment of research and the detriment of patients, and really create a series of collaborative research programs across the board, from Alzheimer's disease to epilepsy to depression to brain injury, and a lot of other areas, where autism, where we, as a routine basis, have psychiatrists, neurologists, neurosurgeons, neuroscientists, rehab medicine doctors working together in the same clinic, in the same research unit. And that really does inculcate everything that we do. That means that as research track residents, you will come into the system and have at your disposal the entire neuroscience community at Mount Sinai. You need not do your project in a psychiatry per se lab. It could be anywhere within the neuroscience community, and I broaden that, including the genetics community as well. We've had many research track residents in psychiatry working with PIs in these other departments who've done fabulously well. And the goal, of course, of the program is to prepare you all to become a tenure track faculty member as quickly as possible. Many research track residents join the faculty immediately after residency. Some take a PG 5-year, that's fine, depends on the research. But the goal is to get you prepared for that faculty position as quickly as possible. That means that one of the secret ingredients of our success is that we do not ask you to be a postdoc, a standard postdoc in a lab. The research projects that you will be taking on will be your projects that you will be groomed to take with you as an independent investigator. We hope it at Mount Sinai, but perhaps somewhere else as well. And we work with PIs, and we will recommend that you identify PIs who are fully familiar with this model that we've perfected here at Mount Sinai. So rather than just joining another lab and acting like a regular postdoc, postdocs today might be 5-6 years long. Well, that's ludicrous. We'd prefer you getting a job and getting a grant, getting on with your careers before you're 50 years old. So the idea would be to have you join an existing lab so that you benefit from ongoing mentorship, but you have greater independence and more ownership of your project than a standard postdoc would find. And we have the funding to support residents in the research years during residency. Those of you who need a PG 5-year, we have funding for that as well. Mercedes mentioned, Renee mentioned R25, T32 grants. We have other funding opportunities available as well. And so we really try to make this as easy and automatic for the research track residents as possible. I think that's all I wanted to say. Mercedes, anything that I should comment on or you'd like me to comment on?
No, I mean, I think you covered everything. And in the interest of time, maybe we can take, a couple of questions for Doctors Nestler and Khan, and then Antonia and I will introduce the programs. So anyone has any questions?
Hi, I have a question. So my name is Chu. I'm an MD PhD student from the University of Florida, and I did my PhD in anthropology. So I was curious to know sort of like how your program approaches candidates from like social science backgrounds.
Yeah, I mean, I'll just give a general answer and Renee can give a more specific answer. We would welcome people with diverse training. We've had people going into psychiatry. Over my career, I've seen people go into psychiatry with PhDs in, microbial genetics, which is arguably a lot less relevant to psychiatry than perhaps anthropology, all the way to social sciences and everything in between. So I think that we welcome a breadth of experience, and you would just have to make sure that the faculty at Mount Sinai realize we don't have an anthropology department. As a freestanding medical school, you would just have to make sure that we have the faculty with whom you'd like to work during your research years. Renee?
Yeah, no, I can certainly acknowledge that. Yeah, definitely. In general, I usually say, doing a PhD is more important than what area the PhD is in, because doing a PhD will form you into becoming a scientist. So that's usually what I've done in the MD PhD students that I have mentored. It's not that relevant, of course. That's not totally true, but it's not that relevant. So yes, for us it would not be a problem at all. If anything, it will be actually a very interesting and important addition to the people that we usually get here and through the program.
Thank you very much. Oh, sorry.
Thank you. Oh, no worries. I was going to ask one more question, maybe, for Drs. Khan and Nestler. Anyone?
Hi, my name is Karen Gomez. I'm an MD PhD student from Columbia, and I'm particularly interested in research opportunities in computational biology, especially psychiatric genomics. And so I was wondering if you could speak a little bit more about the opportunities that are available there.
Yes, so Renee, I mean, you should definitely cover that.
Yeah, we have a very, very strong program. I mean, one of the major leaders is Panos Roussos, who's got a huge program in computation and genetics and in genetics in general. For instance, using the MVP, the Million Veterans Program, where we have access to, and he's one of the main investigators in doing genetic research in 1 million veterans, which is, you know, unique in the world, and also therefore in the US. So we have a very large program. We have a separate artificial intelligence program here at Sinai. We have computational psychiatry, a center of computational psychiatry within the Department of Psychiatry. So there's a huge amount of possibilities to do exactly that kind of research.
And we also have Allison Goat is our chair of the Department of Genetics. She's a neurogeneticist. Her main syndromes of interest are neurodegenerative disorders, Alzheimer's disease and Parkinson's disease, but she's also has a long track record in alcoholism genetics. And she's an active collaborator with many of the leading folks in Renee's department. We're extremely strong in genomics and in computation as well.
Thank you.
Okay, so in the interest of time, we have to move on to introducing the program. And I think Dr. Scan and Nestler will probably leave soon, but we'll connect you, you know, with any applicants that are interested in your labs. So thank you for joining us. And Antonia, do you want to go next?
Thanks for having us, Tony. And any of you have any questions for Renee or me, just reach out to us directly. Easy to find by email. Bye, guys.
Yep.
All right.
Thank you both
Bye.
very much. So I'm sorry I had a patient issue that left me just a few moments late. So I'm sorry that I joined a moment late. I'm Antonia New. I am the program director of the residency, and together with Mercedes, who's the associate program director for the physician scientist track, I run that track with her. So I'm an unusual program director in psychiatry because unlike other program directors in the country, actually, I've had a long research career. I've been federally funded. And so I think that for 20 years, spent my time in a MyRec with my sort of hard money line to do research and investigating the neurobiology of borderline personality disorder using a variety of techniques. And so I am unusual in that I think my own research career has allowed me to understand what it takes to be successful as a research scientist in psychiatry. And I think, in many programs, I think you'll find, and I never besmirch other programs, there are lots of good programs around the country, but in many programs, you'll find a tension between the program directors and the research mentors because they're sort of fighting over time. This is not the case in our program. I understand what it takes. We have a flexibility to create a program, even shifting time for doing one's requirements. I make sure everybody coming out of our program is a well-trained psychiatrist who's met all the ACGME requirements for board eligibility. But given that the programs are individually tailored to help the individuals develop the most successful possible program. And you heard from Drs. Kahn and Nestler that it's extremely, we have an incredibly supportive environment here. We understand that there are certain, sometimes what it takes to do a research career is, let's say, all computational, as we heard. Well, what that means is you need a supercomputer and you need data. Other times, if you're going to work with mice, let's say you might need a technician to help you with your behavioral studies and keep your mice going. If you're going to be working with stem cells, you need to feed your cells every day. So we know that these are the kinds of things that we are aware of, and we aim to make every person who enters our program have the opportunity to be the most successful that they can be and to go on to become independent investigators. So, I know everybody very well in our program. We mentor them very closely. Mercedes and I and Renee Kahn have the T32 grant. We have the R25 grant supporting the program. So we are a very, well-resourced physician scientist training program. So I think the most helpful thing for all of you, I know Mercedes will have many things to add to what I'm saying, and we can introduce more details about our program. I think it's going to be helpful for us to give you a chance to ask questions. But Mercedes, did you want to give a more detailed view, perhaps sharing some slides about what the program looks like, if you like?
Yeah, thank you, Antonia.
Okay,
So
thank
I'll
you.
give the overview first, and then we'll have the joint Q&A. And there's going to be also a breakout room where Antonia and I are going to be there as training directors between 4 and 5 p.m. So I'll go over the details of the track, and then some housekeeping instructions, and then we'll go to the breakout rooms so that you have the opportunity to meet one-on-one with potential mentors, but also come to the training director room so that you can ask us more questions. So, okay, I'm going to share my screen. Okay, so everyone can see the slides correctly. Perfect. So this is where we are, our welcoming remarks by the training directors, and just so that everyone knows what will come next. So we'll have these breakout rooms open, and please reach out to Ms. Cindy Chu. She's our program coordinator, and she's on the Zoom call. So please reach out to her via the Zoom chat if you have any issues moving from room to room when we open the breakout rooms, right? Because you should be able to move through them freely. So we're going to have one with the training director, another one with the current trainees. This is going to be hosted by our chief residents for research, Drs. Lou Jean and Brian Size. We're going to have another breakout room for research, for basic research faculty, another one for clinical research faculty, and then empty rooms available for one-on-one meetings. So the other breakout rooms will be groups, but then there's going to be open rooms that you can occupy for a one-on-one meeting. And just some instructions. So if you're a student or trainee, or in general a potential applicant for our programs, I see most of you have done this already, but it's helpful for us if we have your first name, your last name, your training level and school, and if you're faculty, identify yourself as faculty, right? So that when trainees and students are in the breakout rooms, they know who's faculty and who's another student. And this should be, I mean, you should all be familiar by now with the Zoom settings, but you can change your name on the Zoom settings. So now, I think that, Renee and Eric and Antonia have already mentioned a lot of this, but, there are many good reasons why choosing Mount Sinai for our research psychiatry residency or our T32 postdoctoral fellowship, I think is an amazing idea. Of course, I'm one of the training directors, right? So I may be a bit biased, but our departments of psychiatry, neuroscience, genomics are consistently among the top 10 in the country in NIH funding over the past 5-6 years, and we really have many of the most well-funded PIs. We have an outstanding track record in terms of getting our grants funded. What Renee mentioned of 36% success, that's actually much higher than the NIH current pay line, right? The current funding rate. And this is really in no small part due to a really consistent and strong institutional effort to support physician scientists and to support translational research. And I'm not going to repeat what has been said already, but basically most of the leadership at Mount Sinai are psychiatrists, so psychiatry research is very much appreciated. We all know what it takes to train physician scientists. Antonia and I have, I still have my lab, and I do research. I have a funded R01, several other NIH and non-NIH grants. And Antonia had been a very successful researcher and still is a multiple PI on several grants, right, before becoming training director. And this is very unusual. And I wanted to mention, and again, I don't have time to describe all the programs in detail, but one thing I really want to say and put it out front is that we really value diversity, and training at Mount Sinai means being really, in one of the most diverse cities in the country and maybe in the world, right? Like, and the different campuses of Mount Sinai are located in different areas of New York City, right? So we have some of the most affluent zip codes and some very underserved areas that may be, next to each other, right? And diversity is valued really at all levels. I'm not gonna pretend, we are perfect in any way, right? There's a lot of work to do, and we're, striving to improve. But we have a lot of institutional effort and personal effort also to make Mount Sinai and our training programs as diverse as possible. And there are, pipeline programs from undergraduate all the way to postbac, all the way to faculty, retention and recruitment programs. We have NIH funding $16 million to one of our centers, the Center for Scientific Diversity, to hire URM investigators over the next 5 years for the institution. There's a huge emphasis on diversity in neuroscience. And if any of you is interested, I'm gonna put, my email on the chat, and you'll see it at the end of the presentation. So please reach out and I can send you more information about many of these initiatives. And now going back to, why it is exciting to be a clinician scientist in psychiatry, I would say that there's been a lot of research, many research advances that have not yet been translated to the clinic, right? And this is where clinician scientists are really crucial. You can bridge the gap between the basic neurobiology, the basic neuroscience, and the clinical disease. And this goes both ways, right? You see patients, your phenomenology, your psychopathology, you can ask them better questions in your research, and hopefully your research will result in those preventive disease-modifying treatments that we are mostly still lacking in psychiatry. And we're going to talk about 2 programs today. So this is our physician scientist psychiatry residency track. This is funded through an R25, and our residents are also eligible to apply for Leon Levy Foundation fellowships. And it has a separate match number, which is listed here. And generally, this is designed for those who have very substantial research activities prior to residency. In most cases, this means a PhD, but we also consider MD applicants who have substantial research backgrounds or other degrees, for example, an MPH. And we have had this physician scientist track since 2008. I'm actually one of the founding members of the track. I was recruited in 2008 together with Panos Roussos, who's also in the faculty. And we have a lot of experience in training physician scientists. I was in the track, right? So I worked when Antonia invited me to become the assistant training director, and now I'm associate training director. I really knew what it was, right, to be a trainee in the track. And together we have continued to make it better. So I would say that, our graduates really have a great track record in launching successful research careers and getting K awards. So over the last 5 years, we have over 50% of our trainees getting K awards or other career development awards within 2 years of graduation, which is outstanding. And these are some of the financial advantages that any residents in our program would be eligible for. So there's some funding that is guaranteed as part of the R25 or institutional support. And then you're also eligible to apply to the NIH loan repayment program and to the Leon Levy Foundation fellowship. So these are all things that would be that we can discuss in more detail if you have any questions. Our track is designed really to provide the maximum protected time for research that is allowed by the Psychiatry boards and by the ACGME that accredits residency training. So we really want to provide you, right, as much as possible, because we know that staying away from the lab is really detrimental for a research career. And at the same time, you get outstanding clinical training. Antonia and the Associate Program Director for the General Track, Asher Simon, they make sure of that. And as Antonia mentioned, we are also flexible. So this is the standard schedule of the program. You'll see that the standard research protected time starts with a block of time in the second year, and then 1 day, 1/3 of the time, which is like a day and a half a week. Then in the third year, it's 50% research. And in the fourth year, it says 75%. It really can usually be more than that, depending on your interest in any other clinical electives. But this is flexible, right? So if there is a reason why you really need time in your first year, because there's something, right? There's experiments that are ready to set up. This is something that you should discuss with us when you interview, and this is something that we have modified in the past to suit an individual resident's needs. And I'm not gonna go into detail about, the expectations of each year of the track, but basically what I want to focus on is we are our model is a fast track model, and this doesn't mean that we're going to rush you, to write your K award if you're not ready. But this means that we want to make sure that you have the resources and the planning and the mentorship that allows you to do so if you choose to, and if your research goes well. Of course, there can be delays that cannot be, helped. Like, for example, the pandemic for some people, right? That it stopped a lot of the research. But our goal is really to help you launch your research career sooner rather than later, right? So if you can write your K award, in end of your third year, beginning of your fourth year, and then you can have a K award or at least a scored submission by the time you compete for a faculty position, then would be very happy, right? That we don't have to delay your faculty appointment. And if you need more time, we have a T32, and actually we're going to talk about that now, and other postdoctoral fellowship opportunities. So again, we're not pressuring anyone to graduate early into a faculty position, but we're saying that this is actually possible in our program, unlike other places where you're expected to do a postdoc after a research residency. And this is our T32. So it's our standard is 2 years, but there's a competitive extension to 3 for those who need additional time. It's geared to train clinician scientists, so it includes MDs or MD PhDs, but also PsyDs, any other doctorates that you may have that are related to the mission of the NIMH and to psychiatry and neuroscience. And again, with this program, there's also eligibility to apply for the NIH loan repayment program, and there's a structured mentorship, funding for tuition, for travel, there's benefits, housing. And one thing that's important to mention is that we also have fellowship opportunities for visa holders. So those who do not have a green card or citizenship and they are not eligible for the T32 program, we have options for them, including the VA MIREC fellowship. So I've talked about this already, the success of our program. So I'm just going to put my email now on the chat so that I can end the slideshow and look at you. And we can open briefly for just maybe a couple of questions in the large group, and then we're going to go to the breakout rooms so that you'll be able to ask any questions you have for Antonia or for me in the breakout rooms. So any general questions for now? So Alex, are you there? Alex Charney?
I'm here, yeah.
There he is.
Oh, you're here. Yeah, and thank you. Actually, I saw your message on the chat, and that's what alerted me. I was completely oblivious. And then we have Brian Swice, who I see. We have Georges and Alison Waters. Are you there as well?
Yep, I'm here.
Oh, perfect. Okay, thank you. Yeah, I have several screens with, yeah, so so we can go in alphabetical order. So I think Alex, oh, and actually, and Kenny Chan, are you there? Kenny?
Yes, I am.
Perfect. Okay, yes. So I think it's Kenny. Kenny would go first, and he's representing Scott Russo. And Scott Russo could not come today. And actually, we have. Oh, and actually, I'm getting a message that someone has a hard stop at 4 p.m. So those of you who have a hard stop at 4 p.m., let me know on the chat, and I'll make you go first then. So, okay. Okay, so I think there's just one. So Georgios, then if you don't mind, Kenny, then Georgios can go first. And actually, yeah, okay, perfect. So Georgios, do you mind starting?
Hi everyone. I'm Georgios. I did the physician science track from 2016 to 2020. It was a great experience. I graduated from so basically just a little bit of background about myself. I grew up, I was born and raised in Greece, did my up to the medical degree there. And then I came at Mount Sinai to do my PhD, and then the physician scientist track program. It was a great experience. I was here from 2016 to 2020, and I got a career development award and graduated to tenure track assistant professor position, which was kind of like what the program wanted. Also, because this is a program that, at least for the physician science track, tries to graduate people to independent tenure track positions. So right now, I'm assistant professor in the Department of Psychiatry and Genetics and Genomic Sciences, and I'm the director of the Translational Bioinformatics and Precision Therapeutics group at the Center for Disease Neurogenomics. So when I joined Mount Sinai, when I joined the physician scientist program, I took a 180 from what I was doing before. I was working a lot with animal models, but having 4 years protected basically gave me the opportunity to delve a little bit more into computational genomics. So I did my work at Panos Roussos' lab, who was also a graduate of the program, and I learned how to analyze RNA sequencing data and all these things. And finally, I was able to co-develop a method of genomic feature imputation, which then constituted the basis of my K award and half of the research I'm doing now. So my group focuses in translational genomics approaches. I have built genomically based pipelines for gene target prioritization, computational drug repurposing. And now I'm focusing on next generation integration of multi-omics datasets for gene discovery that includes epigenomics and both at the DNA level and at chromatin accessibility and other stuff like that. And finally, the reason why I was hired by the department is precision medicine applications in psychiatry. So I'm using genetic intermediate molecular features to cluster patients into different groups. I'm doing AHR-based phenotype. We're doing heavy machine learning to identify phenotypes and endophenotypes and treatment response and stuff like that. And again, predictive modeling of medication response and side effects. So a lot of machine learning and genomics. So I'm very happy about the growth that Mount Sinai has supported for my lab. Right now, I have a lot of people, and I'm fortunate enough to have even people who have joined from the physician scientist track program, and we're still collaborating. MD PhD students, graduate students, and a lot of bioinformaticians and one senior data scientist. So this is pretty much the rundown of my lab. Funding is from NIMH, NIA, and I'm sure Mercedes mentioned it, but we have Leon Levy Foundation support during residency, and I also got an NARSAD award.
Thank Thank you, Georgios. And this is, so you're a third generation mentor now, right? Panos, you, and then Simone.
Yeah, exactly.
Exactly. Okay. So, so then, Kenny Chan, if you could go next.
Sure.
Thank you.
Hi, I'm Kenny. I am an instructor in Scott Russo's lab, so I'm just going to stand in for him and give a quick overview of the kind of research that we do, and that we try to combine clinical with basic research to investigate these neuroimmune mechanisms of depression, which are really looking at this association between the systemic markers of inflammation or dysfunction of the immune system with major depressive disorder. And
Kenny,
yeah.
sorry, one thing. I see your slides, but I don't see the presentation mode. I see them like open on your desktop.
Oh, okay. Let me try something else. Is that better?
Perfect. Thank you.
Okay. So we try to apply what we call a translational approach to psychiatry, in that there's this challenge of identifying new therapeutic targets for psychiatric disorders. So in collaboration with the Depression and Anxiety Center at Mount Sinai, we've been collecting patient samples, largely blood, postmortem brain samples, or we've been performing imaging studies in patients as well as healthy volunteer subjects. And we use these samples to perform high throughput screens to identify immune-related biomarkers that could potentially be underlying the development of these mood disorders. We then take these biomarkers and apply them to a mouse model of depression that we've developed, which is called the chronic social defeat stress model. We can test if these factors can actually cause or contribute to the behavioral abnormalities that are associated with stress and depression. And the advantage of using these animal models in basic research are that we can then decipher mechanisms that in turn help us inform clinical trials. And then just a very high altitude overview of some of the things that we look at. If my slides advance it. Yes, so some of the readouts that we look at in terms of both in the human and animal models are that we see that stress causes systemic inflammation. What this means is that we've been measuring different subpopulations of immune cells in circulation, and we can see that in both patients with depression as well as in our animal model of stress, there's shifts in these immune cells, as well as increases in circulating levels of pro-inflammatory cytokines. And we also have seen that there's increased permeability of different body barriers, including the blood-brain barrier and the gut barrier. So we've been really looking at these as readouts, and we're looking at how these things can be caused by stress. And in turn, there's a bidirectional communication between the brain and body. We want to know how these immune phenotypes therefore feed back on the brain to guide and shape behavior. And some of the things that we've been looking at, therefore, have been how when the stress breaks down the gut, you therefore have microbes, bacterial products getting into circulation and thereby propagating systemic inflammation, and how inflammation and circulation breaks down the blood-brain barrier, allowing pro-inflammatory factors to get into the brain, altering neuronal excitability, which we can measure directly in animal models, and therefore change reward and social behavior. So this is just an overview of how we've been using samples from patients in the clinic, as well as our animal model of stress, using a team of neuroscientists, psychiatrists, immunologists, bioinformaticians, and together we're hoping to find these immune-related biomarkers of depression. And yeah, that's Russo Lab.
Thank you, Kenny. Yeah, this was a great outline. So we have Alex Charney next.
Is this visible?
Yes,
Okay,
looks
so
great.
my name's Alex Charney. I'm the director of Mount Sinai's Charles Bronfman Institute for Personalized Medicine. I'm a geneticist and psychiatrist by training. I'm a graduate of the program, the research track program. My lab is split into four main buckets, which are represented by the four boxes here on the screen. The first bucket is our bioinformatics unit. I'm the PI of the Mount Sinai Million, where we're sequencing the genomes of 1 million patients in the Mount Sinai Health System. I'm also the PI of a sequencing study in the international consortia that studies psychiatric genomics, where we're sequencing the exomes of 150,000 people with schizophrenia and bipolar disorder. So we do these massive population genetic studies where we try and identify variants that increase risk for schizophrenia and bipolar disorder and other conditions, and then bring those findings back to the patients in the Mount Sinai Health System to try to understand the mechanisms of disease in individual people. So that's our bioinformatics unit. Types of opportunities there for trainees are developing pipelines for cutting edge analytics of genetic data, doing GWAS, doing rare variant genome-wide studies, and stuff of that nature. Our multi-omics unit is centered around a program called the Living Brain Project, which is a study of brain tissue that we collect from living people. As far as we know, it's the largest collection of prefrontal cortex tissue from living people in the world. We've obtained over 600 biopsies of the prefrontal cortex from people who are undergoing deep brain stimulation surgery, and we perform analyses of this cohort that include comparisons of the tissue to postmortem tissue to understand how the gold standard use of postmortem tissue to understand human neurobiology may or may not be limited. We also ask types of questions in this cohort that can't be asked in postmortem tissues, such as understanding the effects of different combinations of medications that people take, the effects of anesthesia, the associations between brain structure and gene and protein expression. Asking a bunch of kind of basic human neurobiology questions in this study, and a lot of the people in my group work on this type of thing. And for trainees who are interested, there's a bunch of opportunities there to learn how to do multi-omics analyses, work with single cell expression data, bulk expression data, and so on and so forth. Our data science unit is led by Isoda Landy. She's a world's expert in machine learning as applied to predicting outcomes in psychiatry in the real world. The type of work that happens in this unit involves the analysis of EHR data to try and understand patterns of healthcare delivery outcomes in our health system as it relates to mental illness. Individuals who are interested in training in this space work closely with Isota and her team, and the types of projects that are happening there are the investigation of clinician bias using large language modeling, large language models, and applying tools such as one of the most exciting things happening there right now is we're looking at the ability of GPT-4 and GPT 3.5 to extract, to bring structure to the clinical notes in psychiatry, which is we never had a tool for that before, and it seems to be pretty miraculous. So that's the type of stuff happening there, and then trying to use that information to predict what's going to happen to people in the real world when they come to us for care. And lastly is our clinical research unit, which is where we try and take some of our findings from the genetics and genomic stuff that we do back to individual patients. And we have a group led by Nicole Simons, who's terrific, and she recruits patients who carry specific genetic variants. They come back in. We do deep neuropsychiatric profiling of these individuals, and we are trying to understand how different types of risk variants, so variants that increase your risk for schizophrenia or bipolar disorder, how these variants present clinically in different ways, even within the same family, causing different illnesses, even though it's the same molecular cause. So this is the type of work that we do there with patients who are enrolled in these big genomic studies that we do. Anyway, that's what we do, and so lots of opportunities for trainees who are interested, and happy to take questions offline for anyone who wants to learn more.
Thank you, Alex. Yeah, great presentation. So now Alison Waters next.
All right, I'm here to tell you why you must train neuromodulation and research with us at the Nash Family Center for Advanced Circuit Therapeutics at Mount Sinai West in Hell's Kitchen. Maybe beside the fact that we're world-renowned treatment center of excellence for non-invasive and invasive neuromodulation and a one-of-a-kind interdisciplinary research center that's very uniquely embedded in a neurosurgical practice. I think the reason is because we all just live and breathe at the cutting edge of brain-computer interfacing in this particular group. So we get to ask the coolest questions and play with the coolest toys. So I thought I'd pick an example for you today, so you could project yourself. And I'm just going to start with Dr. Mayberg, Dr. Helen Mayberg, who's the director of the center, because she just dropped a big publication and is blowing up Twitter, or what was formerly Twitter today, so you can look that up yourself. But she was an early contributor to what we're now accustomed to learning from functional affective neuroscience. She culled some of the first evidence that one region of the brain, the subcallosal cingulate, or BA25, is hyperactive in states of depression, or if you were to induce profound sadness. And you could reverse the hyperactivity with effective treatment for depression. So she convinced a neurosurgeon to borrow a practice from what was then just a neurological treatment for movement disorders, deep brain stimulation. This is a neurosurgical strategy where you actually implant a device in the brain, and it chronically delivers a little bit of stimulation to a place in the brain, a little bit like a temporary lesion. Anyway, it worked. Well, it kind of worked. And that ushered in several decades of research on improving and trying to understand this treatment that has had an incredible influence on understanding of depression and treatments quite broadly. Another big insight arrived about 10 years later when my colleague, Dr. Choi, also at the center, also taking students, noticed that everyone who got better with this treatment had something in common. The act of contact was embedded at the confluence point of these three white matter fiber tracts that sat right in that region. So then it became a fiber tract story. And in fact, it was really convincing when we took patients who didn't respond to DBS, to the treatment, and then moved the therapeutic contact to that location, and they got better. Wow. In fact, as our center has developed the tools and the strategies around tractography and deep brain stimulation for neuropsychiatric disorder, people are getting better faster, they're staying better longer. So, wow, shock, horror, when the randomized controlled trial came out and was halted for futility early in the study because the treatment group wasn't differentiated from the control group, or the placebo group. Why did this happen? I mean, you can imagine how the community, who's seen people with profound depression respond to this treatment, would just have hopes dashed for FDA approval and dissemination of this really life-saving treatment. So why did this happen? I mean, even the authors of the RCT point to the problem of target engagement. Target engagement is where we're asking neurosurgeons to put the device at a spot that really has no anatomical guide point. It's functional. Even these tractography maps are largely statistical. The colors don't exist in the brain, as you know, but we can measure the cortical response to stimulating those fiber bundles. And that's exactly what we've done in my lab. We've pioneered a new way of mapping individual brains. We call it perturbation mapping. So right now, what you're watching are the voltage fluctuations from the brains of 6 different patients. Each of these patients has a stimulator implanted in their brain to treat depression. And this is how the cortex responds when we stimulate at the tractography-defined sweet spot. So similar patterns with individual differences that likely explain that variance we're trying to understand. And if we stimulate in a different location, so that's the gray box, we get a completely different pattern. That's a patient with OCD who's getting stimulated in the anterior limb of the internal capsule. So I am inviting you to join me in my lab, and we're trying to understand what are the biophysical properties that are underlying these patterns. And we're trying to transform this brain readout into a medical assay that you might see in neurosurgical suites across the world. Each one of these people, principal investigators and also clinicians, could have told you a story like that, maybe 3 or maybe 4. Multiple disciplines represented here. On the cutting edge of treatment, both invasive and non-invasive neuromodulation for psychiatric indications like depression, OCD, and Tourette, but also neurological indications like tremor, Parkinson's disease, and dystonia that have a psychiatric profile that we have to manage as well. And then when you're done the day with us, you'll stumble onto the streets of Hell's Kitchen. Has everything you need to take your mind off of your training woes, from Lincoln Center to RuPaul's Drag Race. It's called Hell's Kitchen's being known now as the new West Village, they say, from highbrow to even higher brow. I'll leave it at that.
Thank you, Alison. And this is, I mean, Antonia and I did not mention any of this, but it's wonderful to live in New York City, and our campuses are in very vibrant parts of the city. I want to make sure we have time for our last speaker, and we're running a few minutes behind, but I think it should be okay. So Brian Swice is our last speaker before we reopen the breakout room.
All right, thank you guys. Allison, every time I see those videos, I'm left with a billion more questions. Let's see, I'm trying to share my screen. Can you guys see a slide with just a title up on the top?
Yep.
Okay. Hi, everyone. My name is Brian. I'm one of the chief residents. I'm in my 4th and final year of residency, born and raised in Chicago, and I've been enjoying living in New York. Despite a global pandemic. So I want to tell you a bit about my work. I'm interested in studying how the brain makes complex decisions. And I'm particularly interested in how we can harness the power of behavioral economics to translate discoveries across species. So I want to tell you a little bit about some of the work that I find really fun and interesting and really, really cool. So first of all, what is neuroeconomics? So it's a field of decision science that's focused on trying to understand how the biological properties of the brain give rise to the cognitive biases we have. And I'm particularly interested in how affect and emotions can interact with and influence the choices that we make. Well, so what is counterfactual thinking? Counterfactual thinking is an area of decision science I've been interested in for the last few years that's focused on understanding not only how does the brain represent the decisions we make, but how also the brain represents hypothetical consequences of things we did not do. And that's actually a critical thing that makes us humans. And it's a feature of higher cognition that can go awry in virtually every psychiatric disorder, but we understand very little about how this happens. And one of the reasons I'm interested in this is that it allows us to probe questions in a measurable way, even ways to model across species that can let us study complex emotions in interesting ways. And one emotional construct I've been interested in studying is the concept of regret, which is just one subtype of counterfactual thinking. It's this idea that the consequences of alternative actions you could have selected perhaps would have led to a better outcome. And the idea that we can recognize and process and register that information is the computational basis of this complex emotion, which can not only alter our affective state, but can also alter our future decisions and our future behaviors. And so I've actually been interested over the last few years studying how we can model this across species. And one problem that stands out is, despite being widely accepted that this emotion is detrimental to one's well-being, no description of this emotional construct appears in the DSM whatsoever, and nor is it a defining feature of any psychiatric disorder to date. So some findings just in general in broad strokes is we're starting to be able to get some bits of information that how we register, react to, and learn from regret may be dissociable biological processes that are processed in different circuits in the brain. And there may even be different subcategories of this emotional construct that may be clinically relevant in different ways. So over the last 10 to 15 years of my time in behavioral neuroscience, I've been working with several groups coming up with ways to translate studies of behavioral economics across species. From humans and non-human primates, we have virtual reality-based computer tasks that humans can perform both in the lab and in the functional MRI machines. We've also translated these foraging tasks across species in monkeys that are freely roaming in an open arena on one day, but on the very next day can sit down head fixed and play the human VR versions on a computer. And we've translated these tasks, these naturalistic foraging tasks, all the way down into rats and mice. So what are all these different tasks based in? So essentially we train animals, including humans, to run around virtual environments or real environments foraging for rewards of varying costs. We can vary the costs by different metrics of subjective value, like flavors, and we can also vary the cost of the rewards by how expensive the reward might be to work for it. So I'm going to play a little video clip for you guys. Here's a video of a mouse running around a maze working for rewards. The animal only has a limited amount of time to work for their sole source of food for the day. After that time is up, the animal goes home and we don't feed them for the rest of the day. So they quickly learn that time is a limited commodity and they must budget their time wisely. And the kicker of this task is we play tones for these animals where the pitch of the tone cues the animal on the cost. The lower the pitch, the lower the cost, the sooner the reward will come. The higher the pitch, the higher the cost. And so animals basically window shop and run around this food court, making economic choices, investing their limited time wisely. So you'll see in this video example, the animal will be presented with an offer and they decide to accept that offer based on that pitch of the tone. And after the animal waits for a few seconds and gets its reward, it'll gobble it up and then run down the aisle to the next location where we present it with another offer and they decide that's too expensive. So they run around this food court, spending their limited time making decisions to invest in rewards. You can see here the animal rejected a few offers in a row and then ultimately foraged and found a more affordable option. What we found was that animals get really good at doing this. And in rare circumstances where they walk away from a good deal only to get screwed and burned over on the next trial, do we start to see the biological properties of animals representing missed opportunities. And so we can see that in the brain. So this has led to a series of findings that we've published over the years. Not only can animals across species can register these events, but animals develop strategies to avoid future occurrences of these events. We've targeted different circuits in the brain that we can perturb certain elements of these decision processes using optogenetics and other strategies, and that different drugs of abuse may give rise to unique decision vulnerabilities depending on the drug of abuse that animals were exposed to. We've translated these discoveries across species from rodents to humans, and most recently we've started to study how computational processes related to regret may become dysfunctional in an animal model for mood disorders and other stress-related disorders. So just a little glimpse of some of the new things that I've been doing here at Mount Sinai with several labs I've been working with. With Scott Russo's lab, postdoc Roman Durand-de-Catoli and I have been working closely together, where we can take the brain out of the mice during the task performance, clear the entire brain to make it transparent, and then we can stain the brain, whole brain, brain-wide, for different markers of whatever might be of interest. In this case, we've looked for different markers of cellular activity, so we can get a brain-wide snapshot of all the neurons that were particularly engaged in the task at a moment in time. We can use this brain-wide imaging tools to get a functional map of different circuits that may or may not be involved in different subcategories of regret processing, and use that to then drive further investigation of regions or specific connections we might be interested in studying. I've been working with Denise Kai's lab to learn new approaches in calcium imaging, where we can implant miniature microscopes into the brains of animals while they're performing our task and record from single units. In this case, this is a recording from the hippocampus seen on the right, and those are individual neurons. And then, of course, the ultimate goal as an experimental biologist is then can we go in and experimentally probe these circuits while animals are making choices? And can we augment the weight with which they process and carry the weight of their mistakes into their future behaviors? So we've been studying how these circuits and these processes can be perturbed by stress. We're exploring individual sex differences in regret signatures in males and females, and how these might be linked to neurobiological underpinnings of depression and resilience to develop depressive-like behaviors. So I've been working with several labs primarily here, including that of Eric Nestler, Scott Russo, and Denise Kai. And my plan is to continue this line of work as I transition to independence after residency and beyond. Thank you, guys.
Thank you, Brian. So yeah, so we had many interesting presentations and we're running a little bit behind. So let's open the breakout rooms again and let's join the breakout rooms as before. And I look forward to seeing some of you in the training directors one. So thank you to the speakers, and I know some have to leave. And if you have to leave now, we'll connect any applicants to you via email. So feel free to put your email in the chat or we'll connect them as well. Thank you.